is to express beta galactosides enzyme in Ecoli using bacterial expression system. This enzyme play a role in breaking down the lactose into glucose and galactose in wide use in blue and white screening for molecular cloning. Then the gene of interest which I have selected is lag gene encodes beta galactosides which hydrolyze in lactose into simpler sugar. It is commonly used in a reporting gene to study a gene expression. Then the coming to the plasmid, which I have chosen is PUB19, which also act as a vector. The lactoreturn allows IPTG includes expression of beta galactosides. So the IPTG is a chemical compound used to induce a gene expression in lactoferrin-based system. Then amplification resistor gene for selecting transformation bacteria. It has multiple cloning sites and has restriction enzymes for the selected gene. So we have two restriction enzymes. One is EcoRIGA with GATTC, and the other one is BAMHI with GGATCC. And coming to the cloning strategies, the first one is a design primer. We have a forward and reverse sequence. Here we get a forward and reverse sequence from the restriction enzymes from EcoRI and BAMHI. Using DNA flood, we get this. And the other step is to clone lag gen into the PUB19, and PCR amplification, the lag gene used to design primers and design the digest the PCR product and PUB19 vector with two restriction enzymes of E. coli and BAMHI, then ligate the gene into the vector using T4 DNA ligase. So we have here in the right side steps for ensure dihydration, nutrition and denutrition and aligning extension, final extension holding. Yeah, how much temperature we are maintaining and how much time we are doing it and how many cycles we are going to make for each step. And coming to the next slide, it's transformation of E. coli. So transformation of E. coli is mixing the ligate mixture with compound E. coli DNA5 alpha cells, then perform a heat stroke. The samples which we have got in a previously that will be leaving in a water tub in 42 degrees Celsius for 45 seconds. Then we take out the sample and place them in a petri dishes with which is on LB agar with amplification and eggshell. This LB agar is a nutrition-rich grown medium used for culturing media, especially culturing or growing a bacteria, in especially in E. coli. So then we'll leave it for 1 or 2 nights. Then we can find some colonies formed. So white colonies indicate successful insertion, while blue colonies uh, inact with a lag gene, no insertion. So for cloning screening and verification, we follow the steps for confirming the cloning. Perform a plasmid mini preparation to extract plasmid DNA, then digest the E. coli and BAMHR restriction enzymes to confirm the insertion, then send for Sanger sequence using lag protomer primer with starts with 5 and ends with 3. Then coming to the final step, protein expression and conclusion. In the protein expression includes beta galactosides expression using IPTG. IPTG is a chemical compound used to express gene expression. And the other thing is verifying enzymes by eggs, gel, blue colonies confirmed beta galactosides activity, and then measuring enzyme activity with yellow color changing. And the other one is SDA, SDS page and Western blotting using an anti-beta galactoside antibody. So here in the picture we can see the blue colonies were formed. Those are actively those we can confirm it as beta galactosides, and in the picture below, there are minus O and PG and plus O and PG. So the positive one, which is yellow color, that is measuring enzyme activity through yellow color changes. And the conclusion here we have got is, after doing all these steps and PCR and incubate everything, we have got the result is the project successfully expressed beta galactosides in Ecoli conforming by white and blue colonies, screening and enzyme activity axis. I think I have did a good thing, good job. Thank you.